Every day, we breathe in and out about 20,000 times. But for millions of people living with asthma or COPD, every single breath can be a struggle, with this oxygen deprivation leading to great morbidity and mortality. My name is Zack Grimmett, and I'm eager to share our lab's research exploring how two tiny molecules, oxygen and nitric oxide, work together to help keep our airways open. When you think about breathing, there are two big jobs. The first is getting oxygen into the body. The second is keeping the airways open so that air can flow. Now, a key player in keeping airways open is a protein called the beta-2 adrenergic receptor. I like to think of it as a valve on the airway walls. When this valve is active, the airways relax and it's easier to breathe. This is called bronchodilation. Now, the big question my research tackled was, how do our cells actually control the number and activity of these airway valves? Now, for a long time, people thought this was controlled mainly by a couple of well-known cellular regulators, proteins called GRKs and beta-arrestins. But we discovered that oxygen itself, together with a protein called PVHL, control how many of these valves are around and how active they are. Now, PVHL is a ubiquitin ligase. It acts like an oxygen sensor and helps to break down the beta-2 receptor by a process called ubiquitinylation, basically small protein tags that tell the cell to break down this receptor. This is where nitric oxide comes in. You might know nitric oxide is something doctors use to relax blood vessels in the heart or to help premature babies breathe. But how it relaxes the lungs isn't well understood at all. What we found is that nitric oxide modifies PVHL in a way that prevents it from breaking down the beta-2 receptor by covalent modification of cysteine thiol groups in a process called S-nitrosylation, shown here with SNO, that then recruits an executioner protein called C-Cybl to destroy PVHL. In simple terms, nitric oxide helps to protect and increase those airway valves so that breathing gets easier. We tested this in genetically engineered mice lacking that PVHL SNO site, so they couldn't use this pathway. And those mice ended up with weaker beta-2 signaling and tighter airways. So why does this matter? Well, diseases like asthma and COPD involve narrowed, constricted airways and difficulty breathing. Current treatments focus on stimulating that beta-2 receptor, beta-2 agonists, the standard of care in every hospital in the country. Now, our findings suggest that we might also target PVHL itself to boost these therapies or even to potentially develop new ones. So by uncovering this connection between oxygen, nitric oxide, and airway control, we've identified a new potential way to help people with asthma and other obstructive breathing disorders breathe a little bit easier. Thank you very much for listening.